Welcome to BioBits and Bytes, the deep dive where we break down complex science into fascinating, digestible insights just for you.
Great to be diving in.
Have you ever imagined tiny, living doctors? I mean, not just a pill you swallow or an injection, but actual engineered organisms working inside your body.
Right, custom tailored.
Exactly, custom-tailored to seek out and fix diseases. That might sound like pure science fiction, right?
It really did not that long ago, but it's quickly becoming a medical reality.
It's truly incredible to witness how rapidly this field is evolving, isn't it?
It is. We're moving beyond merely, understanding biological systems to actively designing them, building them.
Even programming them.
Yes, programming them for therapeutic purposes. It's a profound shift.
Absolutely. Today, we're plunging into that groundbreaking world of living medicines.
Yeah, let's get into it.
We're talking about how synthetic biology, coupled with these really revolutionary tools like CRISPR, is fundamentally changing healthcare.
It's not just small steps either.
No, this isn't about incremental improvements. We're looking at paradigm shifts. We're talking fighting stubborn, drug-resistant infections.
The ones that stump conventional antibiotics.
Right. And potentially offering functional cures for devastating genetic disorders.
And even, entirely new ways to target and eliminate cancer. It covers so much ground.
So our mission today is to really unpack these incredible advancements.
Yeah, we want to help you understand the intricate science behind them, see where they're already making a tangible, real-world difference.
And then, crucially, peek into the considerable challenges, because there are challenges.
Oh, definitely. And the profound ethical considerations that, well, they just come with wielding such transformative power.
It's a field brimming with both immense promise and significant responsibility for humanity, wouldn't you say?
Absolutely. Huge potential, huge responsibility.
Okay, let's unpack this. To start, we need to grasp the fundamental building blocks. What exactly are living medicines and these live biotherapeutic products, or LBPs?
Right, the basics.
And maybe, what do you think is the core difference between a regular pharmaceutical drug and one of these living medicines?
That's a great question, and it really gets to the heart of it.
Feels important to distinguish up front.
It is. So, at its core, living medicine refers to therapeutic approaches that use engineered biological systems.
So living things.
Yes, most commonly bacteria, but sometimes even human cells as the primary agents of treatment.
Okay, so not just a chemical.
Exactly. Unlike a traditional pill, which is, a static chemical compound designed to interact with your body's chemistry.
A living medicine is dynamic. It's an organism.
Precisely. It can sense its environment, respond to signals, adapt within the body, maybe even self-replicate.
Like a tiny programmable biological machine or a miniature factory inside you.
That's a good way to think about it, yeah. A mini factory or machine.
So it's not just a molecule. It's a system that's actively doing work. That's a pretty profound distinction.
It really is. And this broad field falls under live biotherapeutic products, or LBPs.
Okay, LBPs.
Now, the term LBP initially encompassed a wider range of products. For instance, we've already seen significant advancements with donor-derived microbiota products.
Like Rebiota and Valsta, those ones for C. diff infections.
Exactly those. They're huge leaps beyond traditional fecal microbiota transplants, FMTs.
Because they're manufactured and tested properly, more controlled.
Right. Controlled manufacturing, rigorous testing, much more standardized and safer than, say, unregulated FMTs.
Okay, that makes sense. That's already a big step up.
It is. But here's where the classification of living medicine is increasingly narrowing its focus.
To what specifically?
To laboratory-defined microbial strains that are actively engineered. That's where the truly cutting-edge innovation lies.
Ah, so not just naturally occurring microbes, but ones we've deliberately designed.
Precisely. We're not just transferring microbes, we're designing and programming them to perform a very specific therapeutic function.
Okay, so back to my original question.
Yeah.
The difference.
Right. A regular drug is a chemical compound or molecule that interacts with the body. A living medicine is an organism that performs a function.
Like that miniature programmable factory or surgeon within you.
Exactly. It's the difference between giving someone a hammer
and giving them a skilled carpenter who knows how to use it and maybe has a whole toolbox.
That's a perfect analogy. Someone who can adapt and build something intricate.
That makes perfect sense. It's like the difference between a static instruction manual and, a skilled artisan who can read, interpret, even rewrite parts of it.
Precisely.
And that brings us naturally to synthetic biology. To me, it sounds like advanced biological engineering, like we're literally designing life. Is that fair?
That's a very fair way to think about it. Synthetic biology is fundamentally about applying engineering principles, design, build, test, refine directly to biological systems.
So taking engineering and applying it to biology.
Exactly. It's about designing and building entirely new biological parts,
Ah.
devices, systems from scratch.
Or modifying existing ones.
Or, just as powerfully, redesigning existing natural biological systems for specific targeted purposes.
Like programming cells, but with DNA instead of code.
You've got it. Think of it like that. Instead of writing lines of code on a computer, you're using DNA as your programming language.
Okay, so wait, let me see if I've got this straight. Synthetic biology is basically taking life's existing instruction manual, the DNA, and rewriting parts of it
Yeah.
to do new beneficial things, almost like a biological operating system upgrade, giving it new apps.
That's a great way to put it, yeah. You've absolutely got it. It allows scientists to give organisms entirely new functions or significantly enhance existing ones.
In ways evolution might never have done, or not as fast anyway.
Right, or certainly not as quickly or predictably. It's about intentional design in biology.
And this isn't just theoretical, right? Has synthetic biology already delivered some real-world wins?
Absolutely. We've already seen its transformative power. For instance, scientists have successfully introduced well-designed metabolic pathways into eukaryotic yeast.
Yeast, like for bread or beer.
The very same kind of organism, yeah. But they've engineered these yeast cells to ferment sugar and, through this process, produce complex opiate compounds.
Opiate compounds, like painkillers
Yeah.
from yeast.
Yes. It's a huge step in drug manufacturing. Could potentially move away from large-scale poppy cultivation, offering a more controlled, sustainable source.
Wow. So instead of growing acres of plants, you could brew these crucial medicines in vats, like beer, but for medicine.
Precisely. Another fantastic success story is artemisinin.
The anti-malarial drug.
That's the one. It's highly effective, but traditionally hard and expensive to get enough of it from the wild Artemisia annua plants. Very low yield.
Right, I remember reading about that.
Synthetic biology overcame this. They engineered microbes to produce it efficiently and affordably through fermentation.
Addressing a critical global health challenge, especially for malaria-heavy regions.
Exactly. Low-cost industrial microorganisms doing the work.
That really highlights the potential for large-scale, cost-effective solutions. Okay, now, to do all this biological engineering, you need tools. And the name that always comes up is CRISPR.
Ah, yes, CRISPR.
We hear genetic scissors, but what does that really mean? How does it work on the DNA?
CRISPR-Cas systems are indeed revolutionary gene editing tools. The scissors analogy is pretty good.
So they cut DNA.
Yes. Imagine incredibly precise molecular scissors you can program to find and cut DNA at specific predetermined locations in the genetic code.
And once it's cut,
Then scientists can remove faulty genetic material, insert new beneficial sequences, or even precisely alter existing ones.
it's an unprecedented level of control over the blueprint of life.
It really is.
Yeah.
Allows us to edit the very instructions that make us who we are or govern how a bacterium functions.
And Cas9 is the big name, the one we always hear about.
That's right. Cas9 is the most famous and widely known protein associated with CRISPR. It's been used in many groundbreaking applications, including that first approved CRISPR therapy we'll talk about.
But it's not the only one.
No, the CRISPR family is actually much larger and more diverse. There's also Cas12a.
Cas12a, less famous, but powerful.
Exactly. Equally powerful, actively being explored, and now being used in human clinical trials, too. That marks a significant first for that specific protein in human treatment. Shows its growing importance.
So it's not just one tool, but a whole toolbox.
Yeah.
Different molecular capability.
Precisely. And then there's Cas3, an intriguing cousin.
Cas3, how's it different? Not scissors.
Well, unlike Cas9, which makes those precise surgical cuts, Cas3 doesn't just cut DNA, it effectively shreds it at the targeted gene regions.
Shreds it, like a paper shredder.
Kind of, yeah. It effectively destroys the bacterial genome in that area. Less like a scalpel, more like a molecular shredder.
Wow, that sounds aggressive, but maybe useful.
Extremely useful for certain things. This destructive capability is being harnessed for specific therapeutic uses where complete eradication of a target, like a harmful bacterium, is the goal.
That makes sense. Total destruction needed sometimes. And I've heard about something even more refined. Base editing. What's that?
Ah, base editing. It's an even more refined version, a remarkable leap in precision.
How so? What's the difference from the scissors?
Okay, so traditional CRISPR-Cas9 creates double-stranded breaks in the DNA ladder, cuts right through both sides,
Right.
which is powerful, but the cell's repair mechanisms can sometimes introduce errors, creating safety risks. Base editing, however, changes single DNA letters or nucleotides directly.
Single letters. Like fixing a typo.
Exactly like fixing a typo. Imagine the DNA ladder has rungs made of two letters, A with T, G with C. Base editing lets you change one letter on a rung, say an A to a G, without cutting the whole rung.
No double strand break.
Correct. That lack of a double-strand break makes it potentially significantly safer and more precise for correcting specific genetic errors when it's applicable, like using a fine-tip pen instead of scissors and tape.
That's incredible precision.
Yeah.
Going from, scissors to a pen. Amazing. Now, besides CRISPR, you mentioned bacteriophages. Phages. What are they and how do they fit in?
Bacteriophages, or phages, are fascinating. They're viruses.
Viruses. Aren't they usually bad?
Well, these have a unique and highly advantageous characteristic. They only attack bacteria.
Only bacteria, not human cells.
Correct. They are incredibly specific to their bacterial targets. A particular phage might only infect one specific strain of bacteria.
Making them highly precise weapons.
Exceptionally attractive therapeutically, yes. In living medicine, they can be used in their natural form to kill specific bacteria, especially antibiotic-resistant ones.
But there's a synthetic biology twist, I assume.
Of course, they can also be engineered. For example, designed to deliver genetic material, like CRISPR components, directly into targeted bacterial cells.
Like tiny Trojan horses.
Exactly, delivering a therapeutic payload gene editors, toxins right inside the target microbe.
So nature's own targeted delivery system, which we can then supercharge. Wow. Okay, that's a perfect bridge to our next section, breakthroughs.
Let's see them in action.
We've got the tools CRISPR, phages, synthetic biology. Let's jump from the blueprint to the battlefield. Where are living medicines making a real difference? Starting with maybe drug-resistant infections.
Absolutely. This is a truly pressing global health crisis. Costs billions, claims countless lives.
And infections on medical implants are a huge part of it.
Huge. Catheters, pacemakers, prosthetic joints. These account for a staggering 80% of all infections acquired in hospitals, healthcare-associated infections.
80%. That's alarming. Why are they so hard to treat?
And they are notoriously resistant to conventional antibiotics, incredibly difficult. The reason is often biofilms.
Biofilms. Tell us about those. Sounds like something out of a sci-fi movie.
Kind of like biological concrete. Think of them as complex protective cities built by bacterial cells.
The cities.
Yeah, they secrete this sticky protective matrix, sugars, proteins, and stick together on a surface, like an implant surface, which is ideal for them.
And this structure protects them.
Exactly. Once established, biofilms create these impenetrable structures. Antibiotics can't get in, even the immune system struggles.
How much more resistant are they?
Bacteria within these biofilms can be 1,000 times more resistant to antibiotics than free-floating bacteria.
1,000 times. It's like they're in a bunker. No wonder they're hard to treat.
It is a nightmare for doctors trying to breach those walls.
So what's this new living medicine approach? How does it get past the fortress walls?
Researchers have developed an experimental treatment specifically targeting these biofilms. It uses modified bacteria, Mycoplasma pneumonia.
Pneumonia. Isn't that usually a pathogen?
It can be, yes, but here it's engineered for this purpose, and the results so far are quite compelling.
How were they tested?
Rigorously. In vitro, lab dish, ex vivo tissue outside the body, and in vivo, living animals.
And it worked.
It successfully treated infections across all three methods. In mice, injecting the therapy under the skin treated infections in 82% of the animals.
82% against biofilms is a really significant success rate.
It is, especially for such difficult infections.
So what's the immediate next step for this therapy? Where do they want to use it first?
Well, this is an important first step. Maria Luch, a co-corresponding author and CSO of Probiotics, mentioned their first aim is treating biofilms on breathing tubes.
Like ventilator tubes.
Exactly. Because M. pneumonia is naturally adapted to the lung environment. She said clinical trials were expected to start in 2023, designed to meet safety and efficacy standards for lung use.
That's a great question. Let's break that down.
Yeah.
How can something naturally found in our lungs, maybe even a pathogen sometimes, be turned into a weapon against superbugs?
Ah, the engineering trick. It lies in sophisticated synthetic biology.
So they disarm it first.
Yes, the strains used here are disarmed and reprogrammed. Instead of causing problems, they're modified to actively seek out and disrupt those biofilm structures.
On surfaces like breathing tubes.
Right. Their natural affinity for the lung tissue means they can deliver their therapeutic payload biofilm breakers, maybe antibacterials, directly and efficiently to the infection site.
Where systemic antibiotics often fail to penetrate.
Precisely. Targeted delivery plus engineered function makes them a potent new weapon.
Turning a potential adversary into a precise therapeutics. Very elegant. Okay, moving from infections to genetics, late 2023 brought huge news.
A truly landmark moment.
The first ever approval of a CRISPR-based medicine. This really feels like a new era for genetic blood disorders.
It was historic, pivotal, akin to the first organ transplant, maybe, the first approval of Casgevy.
And it happened fast, didn't it?
Remarkably fast. From lab discovery to approved therapy in just 11 years.
Wow.
Jennifer Doudna, CRISPR co-inventor, expressed particular satisfaction that this first therapy helps patients with sickle cell disease, SCD.
A condition often neglected, right?
Exactly, neglected by the medical establishment despite its profound impact globally.
That's incredibly fast for such a revolutionary therapy. So what conditions exactly does Casgevy treat?
It's approved for sickle cell disease, SCD, and also transfusion-dependent beta thalassemia, TDT.
Both serious blood disorders affecting hemoglobin.
Severe, debilitating genetic disorders, yes. Affecting hemoglobin, leading to chronic pain, organ damage, reduced quality of life.
And the way it works is fascinating. A functional cure. What does that mean?
It's a powerful concept. It's quite clever in its approach.
Does it directly fix the faulty gene? The mutation?
Not directly, no. It works by inducing the expression of fetal hemoglobin, HbF.
Fetal hemoglobin, the kind babies have.
Exactly. HbF is naturally found in fetuses and very young infants by precisely turning on the gene for HbF in adults.
It can do the job the faulty adult hemoglobin can't.
Precisely.
Yeah.
It effectively performs the job of the healthy adult hemoglobin that people with SCD and TDT are missing or have malformed.
So it's a workaround, not a direct repair, but it alleviates the symptoms.
A powerful workaround, yes. Effectively acting as a functional cure by tackling the root cause of the symptoms. Patients treated show robust, maintained increases in HbF levels quickly.
And people are seeing real benefits.
Absolutely. Inspiring stories like Victoria Gray, an SCD patient, experiencing a remarkable recovery, living free of the debilitating pain crises. Life-changing.
That's just amazing. And this therapy has already gained global recognition.
Yes, impressively fast. First approval from the UK's MHRA in November 2023.
Then the US FDA.
Right behind in December 2023 for SCD, then later for TDT. Also approved in the EU, conditionally in Bahrain, submissions planned or under review in Saudi Arabia, Canada.
Wow. Rapid, widespread approval. Speaks volumes about the potential and the need.
It really does.
So if I'm distilling this, the core insight here is that CRISPR isn't just a lab tool anymore, it's a clinical reality.
Absolutely. A turning point.
Fundamentally alters how we approach genetic diseases, sets a precedent for functional cures, moving beyond just managing symptoms, rewriting the future of personalized medicine.
Couldn't agree more.
But for something so revolutionary, there must be significant challenges. What are the hurdles to widespread access for something like Casgevy?
You're absolutely right. The journey from lab to widespread access is fraught with challenges.
Like what, technologically?
First, technological access. Manufacturing and delivering Casgevy is incredibly complex. Highly specialized facilities, meticulously controlled environments, rare expertise needed.
Not something every hospital can do.
Far from it.
Yeah.
Few locations globally can currently process a patient's cells and deliver this intricate treatment safely and effectively.
And then there's the cost.
Then there's the headline-grabbing cost,
Yeah.
currently priced around $2 million per patient.
$2 million. That's staggering. Even for a functional cure. That creates massive financial access concerns.
It does.
Beyond the price tag, what are the broader societal implications if these cures are only for the privileged few? Health equity becomes a huge issue.
That's a critical question society grapples with. While the lifetime cost of care for SCD or TDT is incredibly high, potentially millions over decades.
Making the upfront cost maybe seem cost-effective long-term.
Potentially, yes, especially in single-payer systems that look at long-term savings. But the incentive structure in places like the U.S. with fragmented insurance, Medicaid, it presents unique challenges for coverage and payment.
Raises profound questions about justice and fairness in healthcare.
It absolutely does. And finally, there are significant safety and patient burden considerations.
What does that involve?
The treatment requires chemotherapy conditioning before the modified cells are infused.
Chemotherapy, like for cancer.
Yes, similar drugs. It's needed to make space in the bone marrow for the new cells. It's tough on patients, akin to a mini bone marrow transplant.
Carries risks, side effects, long recovery.
Yes, serious side effects are possible. This patient burden is a major driver for ongoing research into next-generation therapies.
Focused on improving safety, simplifying the experience.
And crucially, reducing costs to make these cures more broadly accessible.
That's crucial. It's not just finding a cure, but making it practical. Are others working on these next-gen treatments, maybe using different CRISPR tools?
Definitely. The field is buzzing. Editas Medicine, for instance, is conducting phase 12 trials for SCD and TDT using that other CRISPR protein, Cas12a.
Cas12a. First time in a human trial for that one.
Yes. Their approach also turns on HbF, like Casgevy. So far, 17 participants treated, no serious adverse events reported.
And the results are good?
Efficacy appears comparable to Casgevy. All SCD patients free of vaso-occlusive events, TDT patients free of transfusions, very promising for expanding options.
And Beam Therapeutics.
Yes.
Using base editing, right? That sounds potentially safer.
Yes. Beam has dosed its first participant in a phase 12 trial for severe SCD using base editing to turn on HbF.
Avoiding those double-stranded DNA breaks.
Exactly. Seen as potentially safer, could significantly reduce the risk profile. Then there's the UC Consortium.
The nonprofit one.
That's right. And uniquely, they aim to directly repair the SCD mutation itself, not just turn on HbF.
A true genetic correction.
That's the goal. They faced manufacturing delays, but plan to start enrolling patients in early 2025. Another exciting avenue.
So lots of different approaches, all aiming for functional cures or direct repair. What's the ultimate future vision? Simpler administration.
That's the dream, the holy grail. Researchers like Fyodor Urnov envision gene editing moving outside of the realm of the bone marrow transplantation unit.
Becoming just an IV injection.
That's the idea. In vivo editing, delivering the tool directly into the body to edit cells there.
That would make it so much more accessible.
Dramatically. Reduce the need for taking cells out ex vivo, editing them, and the associated chemo burden.
But there's a catch.
The major obstacle for in vivo is quality control. If you modify cells inside the body, you can't easily test them beforehand to ensure everything went right. No off-target edits, etc.
Right, the control goes down, validation is harder. A huge challenge, but the benefits are clear if overcome.
Absolutely. A significant challenge, immense benefit.
Okay, let's pivot to another huge area, reprogramming the immune system. Cancer and autoimmune diseases. First, engineered bacteria attacking cancer. How does that work? Sounds almost counterintuitive.
It's a really exciting development, turning an old idea on its head. New research from Columbia University highlights engineered probiotic bacteria as a novel class of cancer vaccines.
Cancer vaccines using bacteria. And they're personalized.
Personalized, yes.
Yeah.
Not one size fits all, tailored specifically to each patient's tumor.
How do they do that?
Researchers identify unique neoantigens in the patient's tumor. These are cancer-specific genetic mutations, like unique flags on the tumor cells.
Distinguishing them from healthy cells.
Exactly. Then, a probiotic strain of E. coli is genetically engineered to produce large quantities of these identified neoantigens.
Plus other things.
Yes. Crucially, also, other immunomodulatory factor substances that influence the immune system, basically acting like a potent booster to the body's cancer defenses.
Brilliant. Bacteria becoming custom-made, living factories for tumor signals and immune boosters. How do they deliver this payload?
These engineered bacteria have a natural ability to migrate to and colonize tumors. They thrive in the low-oxygen tumor environment.
They actually seek out the tumor.
They do. Once settled, they steadily produce and deliver their payload neoantigens, immunomodulators, directly to the tumor cells.
Local delivery allows higher concentrations.
Much higher than systemic delivery, which would be too toxic. The bacterial vaccine then supercharges and coordinately restructures and activates all arms of the immune system to attack tumor cells.
While avoiding signals that suppress the immune attack.
Exactly. It's like having tiny, specialized immune trainers embedded right within the cancer itself.
That sounds incredibly effective, almost self-amplifying, self-targeting. What are the preclinical results like?
Very encouraging. In mouse models of advanced colorectal cancer and melanoma, this bacterial vaccine effectively suppressed, or in many cases, entirely eliminated primary and metastatic cancers.
Without harming healthy tissues.
Yes, leaving healthy tissues unharmed, critical for safety. It also showed promise in preventing cancer regrowth in cured mice.
Suggesting a long-lasting preventative effect, preventing recurrence.
That's the hope, yes.
But what about safety? Putting engineered bacteria into a patient sounds risky. How do they control it?
A critical concern, absolutely. Safety is engineered into the design. The bacteria are modified to block their innate ability to evade immune attacks.
So the immune system can clear them easily if needed.
Yes, ensures they're easily recognized and cleared if they don't find the tumor or spread too far. The platform also helps counteract cancer's ability to mutate and evade treatment.
How?
By delivering multiple neoantigens simultaneously. Makes it much harder for the tumor to hide or develop resistance. Highly specific and controllable design.
Smart. Built-in safety, harder for cancer to adapt. Is this idea of using bacteria against cancer totally new?
Interestingly, no. It dates back to the late 19th century. A surgeon, William Coley, observed tumor regression in patients injected with Bacteria coli's toxins.
Really? So it has historical roots?
It does. Crude methods then, but bacteria are still used therapeutically today, BCG, for early-stage bladder cancer, for example.
So an old concept, now modernized and made precise with synthetic biology.
Exactly. Researchers are adding more safety optimizations, getting closer to patient testing. The personalization process, tumor sequencing to bacterial strain manufacturing, could be relatively fast.
Truly promising. Now, let's talk CRISPR-enhanced CAR-T immunotherapy. CAR-T is already a big deal in cancer. How is CRISPR making it even better?
Absolutely. CAR-T, chimeric antigen receptor T-cell therapy, involves engineering a patient's own T-cells to recognize and attack their cancer. Approved in 2017 using traditional gene therapy.
Very effective for some blood cancers, right?
Remarkably effective, yes. Lymphomas, leukemias. With CRISPR, the aim is to make CAR-T even more powerful, safer, and crucially, more accessible.
So what were the challenges with traditional CAR-T that CRISPR helped solve?
Most approved CAR-T therapies are autologous, cells taken from the patient, edited, multiplied, put back into the same patient.
Which sounds complex and expensive.
It is, offers long-lasting benefits, but it's expensive, time-consuming, needs specialized facilities, sometimes fails to produce potent cells.
And patients have to wait while their disease progresses.
That's a critical concern, yes. Waiting weeks for their customized therapy.
So CRISPR helps create off-the-shelf CAR-T ready to go.
Exactly. CRISPR is key to developing allogeneic CAR-T cells, manufactured in advance, large batches from healthy donors, available off the shelf.
Could dramatically improve access, reduce wait times, potentially lower costs.
That's the goal.
What's the progress for blood cancers like leukemias and lymphomas with these CRISPR-modified versions?
Very exciting. CRISPR Therapeutics, for example, is developing next-gen allogeneic CAR-Ts targeting proteins like CD70 and CD19.
And they're making extra edits.
Yes, their newer treatments include edits to two more genes, RNase1 and TGFBR2. These act like brakes on T-cell anti-cancer activity.
So they're removing the brakes for better potency.
Exactly. Aiming for better manufacturing and potency. Caribou Biosciences has also shown impressive results with CD19 targeting CAR-Ts.
So what's special about theirs?
They also have a genetic deactivation, a knockout of PD-1. That's a gene cancer cells use to hide from the immune system.
Taking away one of cancer's escape hatches.
Precisely. In a phase 1 trial, 15 out of 16 participants responded. 7 achieved complete remission lasting 6 months or longer, some for 24+ months.
Wow, that's remarkable durability for an off-the-shelf product.
It really is. It received RMAT, Fast Track, Orphan Drug designations from FDA, signals high potential. Caribou plans to move directly to a pivotal phase 3 trial by end of 2024.
Straight to phase 3, that's fast.
Very fast. And Caribou is also starting trials for multiple myeloma and acute myeloid leukemia, shows the platform's breadth.
And there's work in the UK with children, too.
Yes, reinforcing the picture. The UK NIHR Great Ormond Street Hospital reported positive safety results from a small phase 1 trial using CD19 targeting cells in kids with B-cell leukemias.
So collective progress paints a very promising future for blood cancers, potentially transformative.
Truly. Transforming them from chronic conditions to potentially curable for many more patients.
What about solid tumors? Often harder for CAR-T progress there.
Research is definitely ongoing. kidney, gastrointestinal, breast, pancreatic cancers. Still, in earlier phase 1 trials, results aren't widely available yet.
But the potential is there, adapting these strategies.
Absolutely. Adapting them for the unique challenges of solid tumors, like their complex microenvironment.
Sounds like a potential revolution for cancer treatment. But what about autoimmune diseases? Can CRISPR help there too?
This is a truly new and incredibly exciting frontier. CRISPR Therapeutics is initiating a trial using their next-gen CD19 CAR cells for systemic lupus erythematosus, SLE.
Lupus, using CAR-T, first application of CRISPR in autoimmunity.
Exactly. First CRISPR-modified cell therapy trial in the autoimmune space. A significant expansion.
So what does this all mean for someone living with a challenging disease like lupus? This could be a game changer, but how does cancer therapy translate?
It truly could be. While CAR-T is approved for cancer, applying it to autoimmune disorders is gaining huge excitement, building on compelling early academic results.
What's the idea? How would it work?
The idea is that CAR-T could quickly cause a complete, profound depletion of D cells.
The immune cells implicated in autoimmunity.
Yes, B cells are implicated as drivers in lupus, rheumatoid arthritis, MRC. Depleting them could effectively reset the patient's immune system.
Resetting it, allowing it to rebuild without attacking itself.
That's the hope, potentially providing significant therapeutic benefit without needing the CAR-T cells to persist long-term, which is often a challenge for allogeneic CAR-T in cancer.
So it could be a novel, maybe even curative approach for conditions currently needing long-term immunosuppression.
That's the potential, yes. Moving beyond managing symptoms with drugs that have side effects towards a potential one-time reset.
An immune system reset. Mind-blowing, like restarting a glitchy computer. Okay, let's go beyond the major headlines. Other promising applications, chronic UTIs, such a common problem.
Incredibly common, especially affecting women. Recurring antibiotic-resistant cases are a significant issue. Huge discomfort, frustration, healthcare costs.
So what's the living medicine solution here?
Lucas Biosciences developed a really innovative treatment, combining 3 bacteriophages with CRISPR-Cas3.
Phages and that DNA shredding Cas3 to obliterate the bacteria.
Exactly. Designed to specifically attack and shred the genomes of the E. coli strains causing most UTIs. Kills them beyond recovery.
How is it delivered?
Directly to the bladder via catheter. Targeted delivery right where the infection is.
And the trial results.
Their phase 1b trial completed 2021, first ever using CRISPR for infection. First using Cas3 showed safety and tolerability. Initial results indicated a decrease in E. coli levels.
So it looks promising. What's next?
A larger phase 23 trial for about 800 participants is now enrolling in the US and EU, bringing it closer to potential availability.
Fantastic news for such a prevalent issue. What about protein folding diseases like ATTR?
Yes, hereditary transthyretin amyloidosis, HATR, a fatal progressive disease. Single DNA letter mutation leads to a faulty TTR protein,
which builds up in organs.
Yes, as amyloid deposits heart failure, nerve damage. Intellia Therapeutics is investigating a CRISPR-Cas9 treatment.
How does that work?
Delivered intravenously to the liver, the main site of faulty protein production, aims to precisely reduce the amount of faulty protein the body makes, halting or reversing disease progression.
So stopping the factory making the bad product, not fixing the protein itself.
Exactly. Then there's hereditary angioedema, HAE, rare disease, severe inflammatory attacks, swelling, can be life-threatening.
How does CRISPR help there?
Also uses CRISPR-Cas9 tools. The aim here is not to fix a gene, but to break the gene for an inflammatory protein called kallikrein that drives the attacks.
So turning off something harmful.
Precisely. It's an in vivo systemic treatment. Single 5e dose using lipid nanoparticles, LNPs.
Those tiny fat bubbles that deliver the CRISPR to the liver.
Yes, LNPs have an affinity for the liver. Early trials showed remarkable results. Most participants attack-free for up to 13 months. Significant reductions, 64 and 90% in the inflammatory protein.
Suggesting a functional cure with a single dose.
It really might be, yes. Hugely impactful for patients.
A single IV dose for a functional cure. Transformative. What about broader applications? Cardiovascular disease affects so many.
This is where gene editing truly moves towards population health. Verve Therapeutics started a trial in 2022 for familial hypercholesterolemia.
Genetic high cholesterol, early heart disease.
Right. They're using a base editor delivered by LNP to the liver, making a single letter change in the PCSK9 gene.
To lower LDL cholesterol, mimicking a beneficial mutation.
Exactly. While promising, the trial had some serious cardiovascular events, though mostly linked to severe underlying disease, led to a temporary FDA hold over germline editing concerns.
Worrying edits might pass to future generations.
That's the concern. But the trial resumed in late 2023, shows continued confidence, but caution is key.
Absolutely, especially for large populations. And CRISPR Therapeutics is also in this space.
Yes, they initiated 2 phase 1 trials in 2023 for cardiovascular disease. Targeting ANGPTL3 reduces lipids, and LPA. High LPA is strongly linked to heart disease.
And these target common gene versions,
Yeah.
not just rare variants.
Correct. Unlike the Verve trial, these aim to modify wild type or common versions, potentially impacting a much broader population. Maybe 1 in 5 people globally have elevated LPA.
Wow. Fyodor Urnov compared it to statins, moving towards prevention.
He did, suggesting these therapies might move from treating rare genetic diseases to widespread prevention, fundamentally changing cardiovascular risk management.
Huge leap. Rare fixes to common prevention. Reimagining medicine. And finally, type 1 diabetes. How is living medicine approaching that?
A truly innovative approach here combines stem cell biology and gene editing. A phase 1 trial began in 2022, now solely by CRISPR Therapeutics after an acquisition.
What does it involve?
Pancreatic cells derived from stem cells, the insulin-producing kind. Then, these cells are CRISPR-edited to modify immune-related genes.
To stop the patient's immune system from attacking them, which is the cause of T1D.
Exactly. These engineered cells are then implanted in a special pouch in the patient, aiming to provide healthy new pancreatic cells to control or cure T1D without needing immunosuppressants.
Which are needed for traditional islet transplants.
Yes, and this method also has a scalability advantage over donor transplants, which are limited by availability. The trial was put on hold after the acquisition, but is intended to continue.
Here's where it gets really interesting. It's not just headline-grabbing rare diseases anymore. It's a whole spectrum. Common infections, chronic metabolic disorders, heart disease.
Exactly. The sheer breadth is a testament to the technology's versatility and researcher ingenuity. Re-engineering gut bacteria, restoring insulin production, preventing heart attacks. The imagination feels like the limit.
Demonstrates how living medicines could weed into everyday healthcare, moving beyond single cures to comprehensive, preventative, personalized wellness. We've covered remarkable breakthroughs, astonishing potential. But revolutionary tech always has challenges, perhaps even more complex than the science itself.
That's often the case.
Let's start with manufacturing and scalability. What do you think is the biggest hurdle once these amazing treatments leave the lab and need to be made for millions?
That's a crucial question. Often distinguishes a lab marvel from a real-world therapy. The biggest hurdle is taking engineered probiotics, other living medicines, from successful lab experiments to large-scale industrial production.
Maintaining consistency, quality.
Yes. Consistency in yield, viability, quality across millions of batches. Incredibly complex. Far more so than with traditional chemical drugs. We're dealing with living organisms.
Not inert molecules, so it's like running a very precise living farm industrially.
Exactly. Tiny shifts in temperature, oxygen, pH, moisture during production can critically impair bacterial viability or cell function. Directly affects effectiveness, shelf life.
Like growing a garden where every plant needs to be identical, and a humidity shift ruins the batch.
A good analogy. Optimizing fermentation, bioreactors, advanced bioprocessing is essential. A common industry solution, especially for probiotics, is initial overage.
Adding extra to compensate for losses.
Yes, adding extra active organisms beyond the required dose to compensate for potential losses during production, storage, transport, ensures the patient gets the intended dose.
But adds cost and complexity.
It does.
Okay, beyond manufacturing, there's regulation. Let's pause and simplify that again. Regulation sounds like a real headache for these living therapies. How does an organization navigate that maze?
You're right, it's incredibly complex, arguably one of the biggest bottlenecks. The field of live biotherapeutic products, LBPs, especially engineered strains, is so new.
The frameworks have gaps.
Notable gaps. We've seen approvals for donor-derived products like Rebiota, Vausta.
But those were natural processed microbes, not engineered ones.
Exactly. Those approvals don't fully address the unique challenges of engineered LBPs, designed organisms. The existing rulebook isn't quite ready.
So the guidance is lagging.
The FDA's key guidance for LBPs is limited. A single document from 2016, mainly on chemistry, manufacturing control, CMC, for early trials. No updated guidance reflecting the rapid evolution.
Creates uncertainty for companies.
Huge uncertainty. Imagine building a skyscraper, but the building codes keep changing or haven't been written for your new materials. Companies make best guesses, invest millions.
Only to find regulators have different unstated expectations.
It happens. Leads to immense financial risk, significant delays. Promising therapies get stuck in regulatory limbo.
So it's not just bad for companies, it delays treatments for patients. For you, the listener.
Absolutely. Every delay in guidance is a delay getting these treatments out of the lab and into hospitals.
Like building a house without complete blueprints, inspectors making up rules. What about analytical challenges? Proving what's in it and that it works.
Another major hurdle for living systems. Characterizing LBPs for purity, potency, identity is critical but difficult. Traditional analytical methods often perform poorly.
Why?
Biological diversity, complex starting materials, potential strain-to-strain interference in multi-strain products. Developing and validating new product-specific assays is costly, time-consuming,
So you have to invent the test itself, often.
or adapt it profoundly. For microbial ID, just looking at shape or metabolism isn't enough. Regulators demand highly precise product-specific ID, often recommending two complementary methods.
Like genetic barcoding. Or protein fingerprinting.
Yes, 16S RNA sequencing, MALDI, TOF, mass spec.
Uh-huh.
Advanced techniques. Yeah. Then there's monitoring bioburden contamination.
Keeping unwanted microbes out.
Crucial. LVP manufacturing often involves growing organisms, so
Yeah.
clearing contaminants is tricky. Existing guidance is incomplete. Methods lack specificity.
And risk of antibiotic resistance transfer.
A significant safety concern, too, from contaminants to the therapeutic or the patient's own microbes.
A lot to consider. How does an organization navigate this with so many unknowns?
The absolute key is close, early, frequent communication with regulators, FDA, EMA.
Talking to them constantly.
Engaging in dialogue on all critical steps. Proposing modifications to guidance, providing robust data, scientific rationale, validated assays, less submitting paperwork, more continuous collaboration.
So the industry helps regulators write the new rulebook in real time.
In many ways, yes. It's expected LBP regulations will follow patterns seen with gene and cell therapy specific regulations due to novelty. Requires collaboration, industry, academia, agencies.
Need to expand guidance, develop rules for later stages, commercialization, learn from recent approvals.
Exactly. Workshops, educational programs to build consensus. It's a dynamic landscape demanding adaptation from everyone.
Diligent work, essential for safety and getting therapies to patients. Which brings us to safety and ethics. The double-edged sword.
Indeed.
How do we ensure these powerful technologies are used for good, not harm? What does playing with fire mean biologically?
The core ethical dilemma. First, participant safety in trials. Despite rigorous testing, trials carry risks. We've seen severe adverse events stop trials. Patient monitoring safety protocols are critical.
Every patient is a pioneer. Their well-being paramount.
Absolutely. Then there's biosafety preventing unintentional harm to patients, the environment.
Synthetic biology can create more dangerous pathogens.
It has the capability, yes, if not handled with extreme care, preventing unintentional exposure or release is vital.
Concerning examples where risks seem high.
Recreating known pathogens, making existing ones more dangerous. That novel COVID strain made in a Boston Uni lab.
Fusing Omicron spike with the original strain.
Yes. Resulted in a synthetic strain 5x more infectious than Omicron, 80% mortality in mice.
Chilling. Truly highlights the power.
Experts called it exceedingly unwise, playing with fire. What's concerning is the knowledge for this is increasingly available, even outside highly regulated labs, raises accident concerns.
That is, playing with fire biologically. And beyond accidents, there's malicious use. Biosecurity.
The other critical side. Modified pathogens in the wrong hands could be used for bioterrorism, incalculable hazards.
And the DIY biology movement. Biohacking.
A unique dual challenge. Democratization of tools, knowledge, can foster grassroots innovation like that low-cost mobile malaria test kit Emplino.
Developed by a DIY biologist.
Yes, but many practitioners operate outside traditional safeguards. Lack formal safety training, systematic knowledge, oversight.
Innovation on one side, potential danger, and lack of oversight on the other. Difficult balance.
Precisely. Absence of specialized laws, Limited oversight raises concerns about misuse, like gene editing outside regulated labs. Why robust ethical governance is essential, guided by 4 core principles.
Okay, let's break those down. First, human-centeredness.
Valuing human life, health, well-being above all. Safeguarding patient participants' safety and rights. Respecting autonomy via informed consent, upholding dignity, ensuring equity.
Enshrined in guidelines like Declaration of Helsinki.
Yes, and national regs like GCP. Ethical review committees need to be interdisciplinary scientists, doctors, bioethicists, lawyers, sociologists, public reps, religious figures, holistic oversight.
Critical. Next principle.
Non-maleficence. Do no harm. Most fundamental. Aims for minimal, reasonable, or morally permissible harm. Acknowledges some risk might be inevitable.
But requires weighing harms versus benefits. Choosing lesser harm.
Rigorously, yes. Human challenge trials, for example. Only ethically reasonable if risks are transparent, minimized, truly informed consent obtained. Responsible risk management.
Then sustainability. Long-term implications.
A long-term principle. Harmony between humans and nature, meeting present needs without compromising future generations, ecological, economic, social sustainability.
Requires public understanding, trust, stakeholder involvement, ensuring today's solutions don't create tomorrow's problems.
Exactly. And finally, reasonable risk control. Practical application. Precisely. Mandates robust measures to reduce, eliminate risks, keep them acceptable, identifying hazards, operable management plans, advanced risk control tech.
Like genetic kill switches.
Yes. Dynamic, continuous supervision. Proactive, systematic approaches to anticipate and mitigate risks constantly.
So how do these translate into practical recommendations?
Strengthening ethical review interdisciplinary committees. Continuous researcher training, promoting policy for private amateur research, formal community labs, registration, public communication,
Improving legal safeguards.
amending laws for synthetic organisms, balancing safety with innovation, avoiding a chilling effect, and enhancing technical biocontainment.
Building safety into the organisms themselves, not just relying on external fences.
That's the goal, making them inherently safer. Comprehensive management of profound power.
Okay, finally, the horizon. AI. Personalized medicine, beyond. What does this mean for you, the listener? Medication literally alive and tailored just for you.
Precisely the direction, rapidly accelerating. AI and machine learning are profoundly enhancing probiotic development, helping predict complex behavior, interactions with the microbiome and host. Allows optimized design, predicting side effects, enabling highly personalized approaches.
AI predicting how living medicines act in your body.
Exactly, data-driven precision. And beyond current CRISPR, advances like prime editing offer even greater precision.
Enabling enhanced functionality, fewer off-target effects.
Significantly minimized off-target effects. Integrating these advanced tools yields next-gen living medicines more robust, efficient, aligned with true precision medicine.
Leading to personalized probiotics, the ultimate goal.
It is. Tailored therapies customized to individual health profiles, unique microbiomes, personalized probiotics targeting specific conditions, overcoming limitations of generic ones.
Because generic ones don't work for everyone.
Due to individual variability, no. Machine learning optimizes microbial compositions for precise effects in your body. Aligns with preventive medicine, continuous monitoring, modulating the microbiome for long-term wellness.
So instead of a generic probiotic, one designed for your gut, your health needs, like a tailor-made health blueprint.
The broader vision is that foundational knowledge and tools for one therapy become much faster, cheaper, and more scalable than going back to square one for each new application.
Building an adaptable platform for biological engineering, addressing myriad health challenges efficiently.
Bringing transformative cures and prevention to more people more quickly. That's the inspiring vision.
Today, we took a deep dive into the incredible, rapidly evolving world of living medicines. We explored engineered bacteria tackling biofilms, CRISPR offering functional cures for genetic disorders,
And how synthetic biology opens doors for entirely new ways to fight cancer and manage chronic diseases.
from microscopic organisms battling superbugs to precision gene editing, rewriting the future of health. The potential is truly vast and expanding astonishingly fast.
It really is. Promising a future of unprecedented medical breakthroughs.
As we continue to unlock life's secrets, gain this ability to engineer biology with unprecedented precision, what new frontiers open when our own biology becomes programmable for health, and what responsibilities come with that immense power for humanity.
Profound questions to ponder.
Thank you for joining again on The Deep Dive.